Hi, you guys. This lecture is actually kind of short. It shouldn't be more than about 10 minutes, maybe 15. All right, so we know DNA is a sequence of nucleotides that codes for a functional product. Once we transcribe DNA into RNA, the RNA could become any number of functional products, right? So that RNA could actually be part of a spliceosome. That RNA could be something else. That RNA can become a lot of different things. What we talked about was only that the RNA could become a protein and the steps that it takes to become a protein, right? But there are some other things that RNA can be, and so we'll talk about some of those right away. Yeah. All right. Let's get my pointer out. RNA. All right, there's several types of functional RNA. So remember, a gene codes for a functional product that could be a protein, but it could also just be RNA itself. So once the RNA has been transcribed and modified, so it can be spliced and stuff too, it can be one of several types of RNA. The first type is the one that we talked about. So that's messenger RNA, right, or mature RNA. The M stands for mature or messenger. That RNA is the RNA that is actually brought to the ribosome itself for translation into a polypeptide. Now there's other RNAs. So as soon as it gets transcribed, it maybe gets modified, it becomes a tRNA, and that's it. That's the end of its life. It is a tRNA, and that's its function. Transfer RNA is a short sequence of nucleotides, of RNA nucleotides, that kind of bends and folds on itself into a three-dimensional shape. Because RNA is single-stranded, that means it has the potential to bend, twist, and loop and get some complementary pairing and create regions that are actually double-stranded. So it'll fold on itself and create loops in a three-dimensional structure. Much in the same way that the functional units of amino acids will bend and fold a protein into a functional shape, the order and sequence of nucleotides will bend and shape RNA into a functional shape. And so that RNA could be tRNA. And tRNA's sole function is to carry an amino acid to the ribosome that correlates to the codon that ribosome is actively reading. So the ribosome reads a 3 nucleotide sequence, that's a codon, and it says, okay, this 3 nucleotide sequence codes for lysine, a tRNA that has a little sequence of an anticodon, we'll call it, has an anticodon on it. That anticodon will come into the ribosome and it will actually pair hydrogen bond to the codon on the transcript it's reading and then transfer the amino acid it's carrying to the ribosome so the ribosome can build this protein structure. So we said tRNA has that anticodon, right, that matches the codon. And then that tRNA with the anticodon for the codon has the corresponding amino acid that it's bringing. So it's a shuttle. It's an amino acid shuttle. Then we have rRNA, ribosomal RNA. So once it's transcribed from the gene for the ribosome transcribes into RNA. That RNA then folds into a shape, and yeah, it's got a few small proteins in there too to help it keep its form. But the ribosomal RNA is what turns into a ribosome. So a ribosome is literally made out of RNA, and it translates RNA into amino acids. So it's kind of cool. You get this functional movement of RNA, or you get this functional what is it? Like you get a functional products from RNA, right? And not everything is proteins, right? Some of it is RNA. So the ribosome, the RNA, folds into the large and the small subunits of the ribosome. So the ribosome has 2 subunits, and let's talk about those. So there's a big, large subunit, right? That's this green goblin-looking guy. And then there's a small subunit just below it. So you got large subunit, small subunit. The large subunit is going to have 3 active sites. It's got the A site, the P site, and the E site. The E site you can always remember as the exit site. The A and the P, I think to me it spells ape, APE, right? So you can use that to remember APE. A is the site where the tRNA enters and finds its anticodon, right? That's the A site. So the tRNA are these swirly piled up things, and then they have the amino acid attached to them that are eventually going to be transferred to the ribosome to build this polypeptide. So the tRNA's anticodon matches up to the codon, and then in the ribosome slides one codon over, and that tRNA that was in the A site now moves to the P site and has its amino acid taken from it and placed onto the growing chain of amino acids. So ribosomes are literally doing an endergonic reaction continuously. They're building a polypeptide. They're making a polymer. So they're getting energy from outside to do this process. The P site is where the growing chain of polypeptides is forming, and then it slides one more over. So it's just continually sliding over. So it slides over, and then the tRNA that was in the P site that just lost its amino acid ends up in the E site, the exit site, and it leaves, and it goes off and it finds an enzyme that literally that enzyme's whole purpose is to transfer the right amino acid to the right tRNA. So it says, what anticodon? It's like a checker at a check station or something. It's like, what anticodon do you have? Great, hold on a second. Boom, grabs an amino acid, hands it to the tRNA. tRNA circles back around to the ribosome when it's needed, and and the codon needs that anticodon. That tRNA is going to come in from the outside. It's going to find the anticodon. It's going to bind to it, slides over into the P site, transfers its amino acid to the ribosome. Ribosome slides over one more site, it leaves the exit site. All right, and we get this image that I have. Oh, this image I have right here is the one that's on the test, and so this is the image I'd like you guys to know. You can see the top part, the large subunit of the ribosome, small subunit of ribosome. So they act kind of like jaws, like like mandibles. You've got top, bottom. They bite down onto the RNA transcript. They read it 5' to 3', and that's how they synthesize their sequence of amino acids. In this image, we have tRNAs. In this image, you can see it here, right? So the tRNAs are coming in from the left and leaving out the right. This image, I did this on purpose. I flipped it so you can see that it's still. I mean, I didn't actually flip it, but so. Hold on a second. All right, you see that magic happen? I did it. Okay, this is funny because I've used this slide a lot and I never noticed this, but I had the 5' and 3' ends flipped. Because look, so this is actually, so this is flipped, like I was saying. If you go back to the previous slide, right, this is the A site is on the left and the E site's on the right. So the tRNA is coming in from the left and leaving out the right side. It's doing the same thing except it's flipped. So they're coming. The A site is on the right and the E site is on the left. And so I did that on purpose because you have to remember everything doesn't work from left to right in the cell, right? You could be looking at it. I could be looking at it like this, or I could be looking at it like this. So in one way, it's going to look like it's translating to the right. The other way, it's going to look like it's translating to the left. But nothing's changing. It's just your perspective shifted, right? You're looking at it from a different angle. Okay. So anyways, the P site in this one, or the A site, is on the right. So the tRNAs with the amino acids attached are going to come in with their anticodon. They're going to find their codon. They're going to find the codon they match up to. That happens in the A site. Then the ribosome shifts one codon to the right. And that tRNA now ends up in the P site where it can transfer its amino acid to the ribosome that then, through an endergonic reaction, will bond it to the growing chain of polypeptides. And then the ribosome is going to shift one more unit to the right. And the one that was in the P site will leave through the E site without an amino acid. And then it'll travel back out into the cytoplasm, and an enzyme will connect it to its corresponding amino acid, and it'll come back around and do the same thing over again. So it's this shuttle. It's this shuttle of amino acids to the ribosome. Cool. All right, so let's talk about translation itself. How do we determine what amino acid corresponds to what codon? And so what I have here is a codon chart. So this works, I know it looks like a lot, but we're going to work through it real quick. What's a codon, right? So a codon is that 3 nucleotide sequence that codes for an amino acid. So that means that there's going to be 3 positions. There's going to be the first nucleotide, the second nucleotide, and the third nucleotide. So the codon chart is read like this. The first position, right? You ask the question, right? What nucleotide is in that first position of the codon? Is it a G? Is it a C? Is it an A or a U? So you read that first position and you say, okay, maybe it's a G. Then you go to the second position. So you go one more concentric circle out, and you go to the second position, and notice that for G being in the first position, there are 4 options for the second position. So does the codon read GC something else? Does it read GU something else? Does it read GA something else, or GG something else? So let's say the codon is just GGG. So we got first position is a G, second position is a G, third position is a G. So that codes for glycine. glycine. So G codes for glycine. Guanine codes for glycine. So let's ask this question. For the codon AUG, right, what does it code for? A is the first position. U is the second position. I kept giving it away here. A is the first position, U is the second position, and G is the third position. So that codes for methionine. And AUG is always the first one. It's the start codon. So what you can do in the DNA, which is really fun, and occasionally you'll find a false positive, if you will. In the DNA, you can actually scan the DNA if you have sequenced the genome. You can scan the DNA and look for ATG, going from 5' to 3' direction, ATG, and that signals that maybe that's a start codon, right? Because in DNA it's going to be an ATG, in the RNA it's going to be AUG, and AUG is always a start codon. So that's one way that you can find genes if you've sequenced the whole genome for an organism. You just do a quick heuristic search and look for ATGs and then mark them, and then you could say, okay, does this make sense? Is there a stop codon after this start codon? And there's algorithms and heuristics you can do to figure that out. For the codon UAG, right? What amino acid is that codon code for? So U, first position, A, second position, G, third position. Goes for a stop. So it doesn't code for an amino acid, it's a stop codon. So what it does is it stops the ribosome from translating the poly A tail. Consider that, right? Because there's still that A. Because I mean, there might be enzymes eating up the poly A tail, right? Heading for the actual, juicy part of the recipe. And so if there's all those A's tagged on the end, how does the ribosome know when to stop translation? Because otherwise it would just make a whole bunch of AAAs. So look here, what would AAA be? Lysine. So you could get like a polylysine tail on your amino acid if the ribosome doesn't stop translating. So there's a stop codon, and that stop codon has a termination factor that binds to it. So the termination factor is just like this little protein or RNA thing that comes in. It has an anticodon. It reads the stop codon. The anticodon matches up to it. And that termination factor separates the large and small subunits of it opens up the jaws of the ribosome, and it lets go of the RNA transcript and floats away. All right, so let's just break it down into steps real quick. tRNA has an anticodon for the start codon AUG. So it's going to bind, that tRNA is going to bind to the codon AUG, and it's going to bring methionine. The smallest subunit of the ribosome is going to come underneath the RNA strand and click it into place. That anticodon, that AUG anticodon, which would read UAC, right? The anticodon would read UAC, comes in bind, small unit comes in, crunch. The large and small subunits meet, and that AUG is in the P site. Now the new tRNA is going to come into the P site, and it's going to shift over one, and it's going to start to make it. So large subunit binds around it, begins translation. New tRNA molecules are bringing amino acids to the ribosomes. Ribosomes move down the mRNA's transcript, and they're going to translate its sequence into a polypeptide. So we can ask this question: where do we find ribosomes? Where do we find ribosomes at? And let's take a look at this image, and the ribosomes can be found in two different places. Okay, most of them are going to hang out in the rough endoplasmic reticulum. So that's the endomembrane system that surrounds the nucleus. So once the RNA transcript is made in the nucleus and modified and has all that post-transcriptional modification done to it, it then gets shuttled through the endoplasmic reticulum until it comes through a pore, and the ribosome jumps right on it and starts translation. So you've got some in the endoplasmic reticulum. That's why it looks rough. You can see that if you looked at this under a microscope, it would look like there was abrasive material around the endoplasmic reticulum. And then some are just floating around on the cytoplasm. So if they come across an mRNA transcript that has the proper GTP cap, they'll just start translation of that. They'll start translation of that transcript. Okay, so this slide breaks down everything that we've talked about for the last two units, for the last two. All right, so let's start at the beginning. Yeah. So RNA is transcribed into DNA up here. There we go. Or RNA is transcribed from the DNA. So RNA is transcribed from the DNA. Then in eukaryotes, that RNA is modified. So remember, it's going to get the introns spliced out, exons glued together. So RNA processing is what it's called in this slide. It gets a GTP cap added onto it. It gets a poly A tail added onto it. And then that mRNA is taken out to a ribosome. And the ribosome is going to initiate translation at the 5' end of the mRNA. So that's over here, right? And this one is just falling off because it got to the stop codon. So it starts up here. And then it starts to assemble a polypeptide. The tRNAs are out here in the cytoplasm are bringing the proper amino acid to the corresponding codon. And then finally it keeps sliding down and reading and making this amino acid. And then it gets to the end, and that termination factor is going to separate this large and small subunits from each other. And then this process repeats endlessly until enzymes have eaten up the part of the RNA that is part of the recipe, and then it stops making proteins. And that is translation. Hope you guys enjoyed it.